Thank you. A bit of disappointment. I am not taking on the industrial aspect of the research. That is more of a Rukmini to take it on. Whatever I'll be presenting today is part of academic research that I did during my postdoc and more so when I was at IIT Madras. I moved to Vantage 1 year back. So I have probably a couple of slides on the industrial aspect, but more so in the context of academic research. So that's what you will be hearing from me today. So as the title says, it's about the genome scale metabolic modeling for cancer. Majority of the talk will be on how to build a genome scale metabolic model, more so because we have like tremendous amount of data, be it clinical, be it physiological, be it omics. So everything put together, you can integrate it at the level of a genome scale metabolic model, since it is derived initially from the genome annotation data. That's the reason it is called as a genome scale metabolic model. Please see to it, this is only talking about the metabolic aspect of the disease. However, when we talk of disease, we do not see only metabolism. You have to see it in a totality. You have to go beyond metabolism. That's the reason I have few slides on where we talk about how do, once we know the impact of metabolism on cancer, can we go beyond metabolism? That's where you will see some slides on the industrial aspect of the research. But more so, I'll talk about what is the role of metabolism in cancer, how has the view changed with respect to metabolism. Moving on to the one of the most used metabolic networks for cancer studies and various other disease studies is the human metabolic network. Am I audible? Yeah, okay. Then I'll talk about the various strategies of building a genome scale model. Either you can build it from scratch, that's the more labor intensive process, probably a period of 6 to 9 months. Cut to, you also have a lot of data where you can also take the top down approach using data for building your model. And then I'll give you certain examples of how you can use the available omics data, publicly available omics data on cancer to build omics informed models and make certain interesting conclusions out of it. Yeah, so that's how the talk is outlined. So the first thing is, what exactly is the impact of metabolism in cancer? What we have seen over the years is the cancer cells, they rewire the metabolism. That means they keep on changing the metabolic type. What I call is metabotype, or the metabolic phenotype of the cancer cells keeps on changing depending upon the tissue environment that it is being exposed to, right? So what is the major driving factors? We're yet to know, but some studies do claim that because of the nutrition and other resources that the cancer cells derive, mostly from the healthy tissues or from other surrounding tissue, they have to adapt in order to accommodate to the increasing demand. They have to continuously grow and proliferate. That's why they keep on adapting, they keep on changing, that there is a severe dynamic behavior of the cancer cells that we are exposed to. And then of course, cancer cells utilize a range of feed-forward and feedback loops in the healthy cells to convert it into a cancerous type. So I'll give you one example where you see this is a normal cell, and these are the metabolic pathways that are operating in the cytoplasm. This is your mitochondria. Anything that you see in red are all those genes/enzymes. Genes will encode for enzymes. Enzymes are proteins that are either associated with cancer, either upregulated or downregulated in cancer, or they could be the targets for the typical cancer. This was a review paper from where I picked up this figure. Very interesting review. This was actually one of the landmark papers where they talk about at length about how you can explore the metabolic aspect of cancer in order to come out with novel therapeutics for killing the cancer type. Now, when I said in the beginning, when we talk about any kind of disease, we are not only interested in the metabolic aspect, we need to go beyond as that. So one thing, even if we are saying there's certain metabolic pathways, in this, it is the glucose transporters, the glucose getting in, and then finally you have the glucose oxidation. In order to enable this particular metabolic pathway, you have certain upregulation as well as certain downregulation of signaling cascades that is operating above it. And that's where you see these kind of signaling cascades that are operating. So these dotted lines are actually the signaling cascades. So the cancer cells majorly modify these, and then because of the modification of these signaling cascades, you see a major impact on the downstream metabolism, right? Okay. So what has majorly changed what we thought about the cancer metabotype to what it is currently now? The focus on the metabolic aspect of the cancer cell arose because of the something called Warburg effect. What they used to see that glucose, in order to provide energy for the cancer cell to survive and proliferate, it used to undergo anaerobic oxidation, meaning it used to give out lactate. So there was this concept that either it can go only glycolysis pathway or oxidative phosphorylation. Glycolysis happening in the cytosol, oxidative phosphorylation happening in the mitochondria. So either of these two has to get turned on or turned off. However, if you see the current view, this is actually not on-off switch, but rather both can coexist. Again, it highlights how heterogeneous is the cancer metabotype and how we need to exactly take advantage of this heterogeneity of cancer in order to come at novel drug targets and therapeutics. So this was just an introduction on, so this is basically what we had believed before, that either you can operate in red or blue, but current concept is you can operate either way. Both can coexist in the cancer cell. There is no on-off switch, but rather there is. What we have also seen, as I'll show you some examples down the line, typically in the model for cancer, colorectal cancer that we developed, we exactly saw this phenomenon as well. Okay, so we move on to the next one, which is the human metabolic network. So the concept of the human metabolic network was, the first metabolic network was basically developed for microorganisms. From the lower organisms, we evolved to form the metabolic network for the human. So most of the reactions that you will see for the metabolic network, when I say metabolic network, what I mean is the components are basically the genes, the enzymes, the proteins, the metabolites, and the reactions. These are the chief components of the metabolic network. So most of the reactions were actually based on that of the E. coli metabolic network, right? Okay. So this is the most comprehensive metabolic network, which we call as the Recon 3D. Sorry, that's the most comprehensive. As you can see, we have a bunch, well, bunch as in we have close to 13,000. If you look into the current, this was the one that is published in 2018, which is maintained on a platform called Virtual Metabolic Human, the VMH. The current content goes beyond 13,000, and the number of metabolites are close to 5,000. The way it was built was integration, a lot of omics data. There are some slides where I'll give you certain concepts of what exactly is omics data, how you can integrate it. But right now I'm showing you the resultant of integration of all of these data types onto a metabolic network for human. This is a very generic network, meaning it doesn't specify the organ or the cell type. Any cell type, any organ within the human that has the generic framework of the metabolism is captured by these models. Okay. So moving on, the way we built the model was majority of the, okay, so I'll give you just a history of it. The first version was Recon 1, published in 2007. Then we had Recon 2, and now we have Recon 3. So we are building upon the existing ones. The major addition for coming up with Recon 3D came from the metabolomics data. If you have questions, that will be answered at a later stage. I'll give you the details of what exactly is metabolomics, but I'll just give you, you get a set of metabolites that are being flushed out from any kind of cell. So that means when we are saying this model represents any given generic cell of the human body, this particular model should also secrete these kind of cells. So we arrived at a bunch of data sets publicly available, which were not captured by the then Recon 3D. So we started integrating these data types onto the model. This was a very top-down approach, sorry, bottom-up approach. We had to study a bit of literature in order to integrate. So the basic pathway for integrating the metabolomics data onto a existing network using the bottom-up approach is you acquire the data, you process the bit of data so that your data is kind of matching up to the model inputs. Then you add it to the network. You have to have both the synthesis and the catabolic pathways of this particular metabolites that are coming into your data set. And then you do a series of network function tests. Most of these are again explained at the end of the presentation. Then from single metabolites, you map on to the reaction. These reactions are then included in a set called pathways, and bunch of these pathways give you the human metabolic network, which is pretty huge. As I said, currently it's more than 13,000 reactions and close to 5,000 metabolites and some close to 3,000 genes specific to only human. Okay, so what we achieved, we did a lot of these additions manually then. So these were total number of new reactions were formulated, new metabolites were included, and certain genes were also included. Then once you have this particular framework, you check for the functionality of the model. How good is your model behaving to capture the human phenotype? You formulate your objective reactions, you formulate a lot of these called the metabolic function test in order to see whether this particular model is capturing the behavior or not. So this part was completely automated, whereas this was a bit of manual exercise that we did. Now, once you have the model, before getting into the network function test, you convert your set of reactions, which are basically looking in a form of a map, network map, into a computable model. So this is what the basic steps are going from a network map to that of a model. So more about this at the later slide, but just to show you that this is completely automated. You have to feed in knowledge only at this level where you define your system boundaries. When you take the entire network as a system, you have to define what is your input, what is your output, right? So most of the time the inputs are pretty fairly easy to write in compute. The outputs are something that we are not sure about. Inputs can be your nutrient data or the environment under which the omics data was generated. All of that can be simulated to give you the input type to your model, and outputs, depending upon the metabolic architecture or the type of reactions this particular model has, the outputs will differ, right? Okay, then you formulate a lot of constraints. Most of the constraints that were set for recon were environmental constraints. That means the nutrient media served as the constraints. Then once you have this, you use the principles of linear optimization. You keep on adding your constraints, going from environmental constraints to that of a regulatory constraints and various other types of constraints you keep on adding. Finally, the solution that you get for your objective. Now, this is not multi-objective function. This is single objective at a time. So this is not what we call as the flux balance analysis, one objective at a time. So you can have like production of glucose, right? Synthesis of lactate and secretion of pyruvate. These kind of simple biochemical reactions or objectives can be your model objectives as well. You keep on adding successive constraints, and then your solution would be, what exactly constraints are doing is your solution space is getting bounded. Now when you apply the linear optimization, the one of the optimal solution will fall on the edge of your solution space, and that is what the FBA, flux balance analysis, is giving you the final optimal result. Okay, this is one of the most popularly used model analysis method. There are plethora of model analysis methods. Look into the protocol for Cobra Toolbox, I think published 2020. They have more than 100 model analysis methods, and FBA is, of course, has been used right from microorganisms to industrial level to that of this kind of academic research level as well. Okay, so you try to gain certain insights of the metabolic functionalities of the model. Okay. So to give you more idea of how to formulate an objective, what exactly is a constraint, and why is it biologically important to include the biology into your constraints and your objectives, I have this slide. Imagine your system let's say this is your recon or any kind of cell type for which you're building a model, one of your objective definitely is production of energy from glucose. That's the typical objective where most of the cells will utilize glucose for. So what are the constraints that you will place? Constraints, of course, glucose has to be in there within the system and we need maximum energy, so it has to be an aerobic oxidation. So the second constraint would be availability of oxygen. The third most important constraint and most of the time overlooked is the presence of the membrane transporters. Whether the glucose is there in the system is ensured if there is a glucose transporter to bring the glucose from the outside to the inside of the cell. And then of course you have to ensure that if it is saying in the literature that my particular organism or my particular cell type has complete oxidation pathway for glucose, you have to ensure the model is also capturing this. If not, you have to fill in the gaps. Where exactly is this particular pathway blocked? Okay, so that exactly what I mean by formulating an objective, designing your constraints around it, and then finally obtaining a positive value for the objective function. Okay, so just to give you the takeaway from this important slide is Recon was formulated to carry out for more than, it was 431 when we were designing it. Currently it can capture more than 500 different metabolic functions that the human body can function as well. And then of course, while designing these functions, we were also aware that since we are saying it is a generic model, it captures any given cell type within the human body. That means if you are trying to design certain organ specific tasks, that means production of bile acid from the liver, that's very specific to the liver. Range of transporters that are specific to the small intestinal cells, are these also captured in the Recon or not? So that's why we had devised close to 100 organ specific objectives on which Recon was tested, and it qualified as well. So that means you have to give a bit of knowledge, biologically rich knowledge, in order to formulate the correct objectives and the constraints around it. So once we have the model, we tested it for a lot of metabolic functions, we had to see whether it is capturing any disease phenotype or not. See, Recon was not built originally just to serve as a knowledge base or a library of metabolic reactions. Rather, we also incorporated a lot of structural information for the metabolites, structural information for the proteins. Okay, so one of the biggest advantage, so this is basically, don't get dissuaded by this particular figure, it's just the different steps that were involved in coming from Recon 2, that was the last model version, to that of the current one, which is Recon 3. So what I meant to say was, because we also integrated a lot of structural information, now if you compute a gene deletion or a genetic mutation, you can actually see the impact of this genetic mutation at the level of the proteins. And that's what we say, that this is the biggest advantage of using Recon. You can move from the language of genes to the level of proteins. Okay? And then of course we had certain important genes that were not that well identified by other models. Now Recon was one of the human metabolic models out there. You have different versions of metabolic models for human by different groups. We compared Recon to those. We saw that it is fairly okay in predicting certain genes which are important for certain diseases, while the other models are not doing that. Okay? Metabolic, yeah. No. No, we didn't go to that. We just deleted one particular gene. We did not go on the SNP level as well. Yeah, it was just single gene deletion. And what is the impact of that gene deletion at the level of proteins and then at the level of reactions? And do you see any clinical phenotype? And some of it actually matched to one of this particular cancer type. This is glioblastoma multiforme, and another one was a metachromatic leukodystrophy. Right? So which the only reason I'm showing you was the other models did not capture this, whereas Recon was able to capture. But if you see what I feel, in my opinion, the biggest advantage is to see the impact of genetic mutations at the level of proteins and at the level of reactions as well. That translation was easily captured by Recon, and that you can do once SNPs I would, yeah, because we also have these different levels of information. If I go back, atom mappings, if you could translate that SNP information onto the level of metabolite, which is actually difficult, but if you have that data, then probably it can capture the SNP level information as well. Yes. Yes, only with respect to metabolism. Yeah, which is around 20% of your human genome. We are kind of neglecting the non-metabolic functionalities, okay, which we say can be captured anyways by the other different gene translation, gene regulatory networks or protein translational networks, right? Yeah. Okay. Yes. Okay, so I'll move on to the next one. Yeah, so finally we come down to what are the different ways that you can build a metabolic network. I give you examples of actually the bottom-up approach as well as the top-down approach. We start with the whatever you have, either you start from the literature, which is the bottom-up approach, or you have a humongous amount of data, which is the top-down approach. You can also, it is not necessary that you either use this or this only. Actually, what we end up doing, we end up using both. So it's basically a hybrid technique. Whatever you are using, you first thing that you have is a draft network, which I call as a draft reconstruction. You curate it, you fill in your missing information, and then you convert it into a mathematical model so that you can easily compute it, because networks are flat. You can get a lot of these sorry, topological information. However, if you want to get into the characterization of what can be modified in terms of diet, drugs, reaction level, those kind, you need a bit of a model. That's why you have to convert your flat maps into model format. You do a bit of model evaluation, whether the model is behaving as that of your phenotype, and then finally you do that iteratively and you get your final set of metabolic models. Okay. Yes, yes. Yeah. So, yeah, getting your question. So, the kind of modeling framework on which all of the simulations are done, these are steady state models. Okay, we do. You can incorporate kinetic information in order to capture the time scale of your data. Steady state meaning whatever input you have given exactly equals to that of your output, and there is no accumulation within the cell. Whatever is going has to be utilized and then the remnants are to be thrown out. Okay, so computing at one particular time point. However, if you want to have it at different time scales to see the dynamics, these are not the choice of models that you will be looking for. They can, but not the kind of modeling platforms that I am discussing. There are a lot of these, well, not exactly advanced. There is something called dynamic FBA, flux balance analysis, as a dynamic component of it that you can use. There are various other ODE based techniques that they have integrated with genome scale models to predict the time scale component of the data. You can do that, but that is not what I'm discussing today. Okay, a bit of confusion as to what exactly is network models, metabolism and everything. Okay, so when I say network in this particular discussion today, I mean a metabolic network where your links are your reactions and your nodes are your metabolites. Okay, what you have been hearing in other discussions are probably the ones where you have a gene regulatory network and protein-protein interaction network. That is not what will be covered today. This topic, this particular discussion, only with respect to reactions and metabolites. Okay. So the bottom-up approach is a very lengthy process. Used to be lengthy process. It used to take probably 9 months to 1 year. We have automated most of it. So right now what you see, this 96-step protocol for using on some amount of genomic gene annotation data, building your organism specific or organ or cell type specific metabolic model can be automated using this protocol. And currently I think the time frame is somewhere close to 6 months, given that you are coming with the fully functional flux consistent model, which is matching the phenotype for which you are testing it. Okay. Okay. So Apologies, this is a very text heavy slide. I had to do it because these are certain things which is, I cannot say it cannot be automated. It is slightly difficult to automate, okay? Now with the advancements in AI, with your LLMs available, most of them can be automated, but you have to be the domain expert to decide the model components that finally end up in your model, okay? So when you curate or you rectify the draft reconstruction which is originating just from the gene annotation data, please mind the gene annotation data itself can be full of errors, okay? So each time you are using any kind of data or any kind of well, not omics particularly, but any kind of clinical or physiological information, it is very much specific to the time point on which the data was generated. It cannot, again, I'm slightly not okay with using can never. To some extent, it cannot take the entirety of the cell. In order to get the entire functionality of the cell types, you have to consider different data types, as well as you have to have a bit of domain expert reading the literature in order to rectify all of these steps in order to get a very high quality model at the end, okay? So I think are we giving out the slides and all of these information? Yeah, I can. So just because I do not want to take time in explaining what are these exactly, but if somebody is interested, please feel free, raise your hands, ask questions. But these are very generic things that one has to be aware of and taking note that it is never error once it ends up in the model. Okay, and you have to have or read a bit of literature in order to correct this information that is captured in your model. Okay. Clear? Okay. Right. This is another layer of information that we feed into the metabolic model, which is what we call are the gene protein reaction association. When you are using your genome annotation data, you have to also include what is the protein for which it is encoding. And finally, if the protein is an enzyme, what is the reaction that is getting catalyzed by the enzyme? All of this information is encoded in the form of a Boolean logic using and or relationship. Simple. If you are coming across a enzyme which is a multi or enzyme complex which has got different subunits, and assembly of all its subunits are essential for its functionality, and if these subunits are encoded by different genes, then the GPR formulation will take an AND rule. Why? Because all the genes are essential, all the subunits are essential, finally giving you the functional enzymes which will catalyze the reaction. However, if you come across an isozyme, something like this, which is a I'll give you an example of glucose phosphorylation, which can be done by glucokinase specific to glucose, but it can also be done by an enzyme called hexokinase, which has got a specificity for a number of hexoses or C6 sugars. Okay? In that case, which is an isozyme, you will feed an information at the level of OR. Okay? So this is your simple logic of using and or relationship, and because we are integrating a lot of these information, you can at the end actually incorporate and integrate a lot of these omics data types onto these models. Okay, just to showing you the application side of the model as well. Okay, again a very controversial topic, how to formulate the objective function. In case of lower organisms, it's pretty simple. The biomass objective function, which means that the cell has to grow, it has to live in order to perform its action, right? But when you come to multicellular organisms like us, living or growth of the cell is not the primary objective of our cells. Rather, it is the maintenance function. Sometimes not even growth, not even maintenance, something completely different from what the phenotype is, depending upon the external environment that this particular cell type is subjected to. So how do you design such objective functions? That is what this particular will tell you. Well, this particular paper gives you an example of E. coli. Even designing the objective function or the biomass objective function for E. coli is slightly tricky. You have to consider a lot of factors in order to form a new cell. What are the essential metabolic components that should go right from the level of micro to that of the macro levels information? That is what is fed, if you see here, We start from protein, RNA, DNA and everything. We also move on to the level of amino acids, to that of fatty acids, to that of sorry, nucleic acid bases. So both at the level of macro and micro level information is fed to formulate your objective function. Right? Okay, and these are the different kinds of constraints that I was talking about, and most of the flux balance analysis models operate under the steady state formulation. And also what we insist that all of your reactions are mass and charge balance. That means mass cannot be formed out of nowhere. Okay? Whatever has gone in, it has to come out, and there is no accumulation within the cell. I have already discussed these, but just to give you again a head start as to what are the different kinds of constraints that you can apply for these different kind of models. Okay. Yeah, this is the final stage. I know it is a kind of labor intensive, lot of things that you have to worry, but this is only for bottom up reconstruction or bottom up modeling approach. Whenever you are getting the final model, you have to test it. You have to see whether there are gaps. In case you have to retain gaps because knowledge is not available at that particular stage, so you just write it off. However, for other things where the knowledge is available, you compute something called the topology of the network. At that level, you can see the gaps that can be easily covered. Okay? And then you do a series of objective functions that your network is tested for. And most of the time, the transport reactions are missed out. Okay? This is the stage where you add a bit of transport information and then to give your final flux consistent model at the end. Well, when I say flux, what I mean is let's say a reaction A getting converted to B, the flux is the millimoles per gram, millimoles gram of B that is formed from A per gram dry weight per hour of the cell. Okay, that's the unit of the flux, and that's what we compute, and that's what I say is when I say flux consistent model, that all the reactions in your model should have a positive flux value. Okay, that's what you're looking for. Well, the reactions are pretty dynamic. It can be ranging from milliseconds to that of a minute. But we do not incorporate the timing information at this stage at all. Yeah, biomass is tricky because when you are writing your biomass, of course, you're building a metabolic model, but you cannot just think about the metabolic components of the cell. You are also accounting for the non-metabolic components. So the paper that I just referenced to, they have given you detailed explanation as to what you should consider when you're accounting for the non-metabolic component that forms your biomass as well. That is also incorporated into your biomass reaction. And if you click on the Recon 3D model and biomass reaction, we have given you 3 different versions of biomass reactions depending upon the environment under which your modeling strategy is getting. To be truthful, this particular Recon biomass reaction was derived from E. coli. We had to make the human conversions, only we had to change the coefficients, and at some time, as in when you are writing your biomass reaction, you have to mention how much of DNA, how much of RNA, how much of protein, and then you have to how many nucleic acid bases, how many amino acids, and then in case of lipid components, what are your phosphatidylcholines. These are very different levels of biochemicals that are there in order to formulate your cell membrane and then for a living cell as well. Okay. So that part was easy. I think the tricky part was to get the coefficient, because it will not be the same as that of the E. coli. So that's what you have to extrapolate. And sometimes there are certain metabolites which are not part of the E. coli at all, but that are very human specific. So you have to get that information. So I think we started out with E. coli, and we read a bunch of literature. Whatever was available, we pulled it into. And then when we tested, we were not able to match it up. I'll give you one example to that of a whole organ resolved model where we did a bit more of studying. We included more human specific parameters. That's when we were able to catch at least the BMR information. That was exactly the same as that of the experimental value. Fine. Yeah, so this ends with the bottom up reconstruction. The more advanced and the more what most of the people are adopting now is the top-down approach. And that's why I give you a set of slides where I'll give you the different algorithms that are available. Actually, the total number of algorithms are huge. What I have on this slide are the first generation algorithms, the basis or the ones which started out the automated reconstruction. What we are using now are the derivations are a bit modified and advanced version of the first generation algorithms. Okay, people also keep using the first generation algorithms only because the mimicking of the data to that on your model is much more higher with the first generation when you move to the currently used algorithms. Fine. So this is one slide just to give you an idea as to what are the different levels of omics. Omics is not just, I think this particular audience is pretty okay with this slide, right? I don't need to explain what exactly is central dogma, your translation of the language from the level of nucleic acids to the level of proteins, and then they finally come and catalyze your reactions. And with each layer of transformation happening, you have the corresponding omics data, but it is only captured at a time point. Okay, so these are snapshots of the functionality of the cell at that particular time scale. Okay, so you can integrate all of this onto a metabolic model, but what you're seeing at the end are the impact of the data at the level of metabolism only. We do not capture at this current stage the non-metabolic functionalities of the cell. This is a slide which I want to show you, only with the the omics. There's one more omics called bibliomics, where you have the lot of literature data coming in. There are different, I think I did not put in fluxomics. You have the transcriptional regulatory information, you have post-translational modification omics, okay, newer kind of omics, thermodynamically constrained omics, right? So all of that can be integrated onto a metabolic model as well as some other biological networks. Okay, and you can use a series of network analysis tools for a number of these kind of analysis. And basically, majority of the metabolic models that I'll be showing here has been used in discovery of the missing parts, okay, and understanding or to get an in-depth knowledge of the biological processes at the level of metabolism. Okay. Okay, so this slide is again a text heavy slide, but I tried to summarize most of the algorithms that are available for integrating different kinds of omics. So the way it works is, what exactly is your purpose? If you want to simply construct a model out of omics, then you can use this set of algorithms. However, if you want to gain a bit of idea of what exactly is my metabolic state of the model or of the data, you can use these set of algorithms. Most of them are actually used synonymously. If you see this one, the advanced version of this is actually the MBA. Again, there are certain point to be noted when you're using certain algorithms. Let's say this one, when you're using model building algorithm, you need to have an idea what are my set of reactions that are definitely being performed within the cell type. That's what formulate your core reaction set. Okay, and most of these algorithms will require this kind of information. So you need to have a bit, along with your data, bit of domain knowledge as well. Okay, and these are the different data types that you can okay, I'll give away the slides. Okay, so I have to like really write it up. Please listen and understand what exactly is the usage of these algorithms. There are plenty algorithms right now. Well, we started out with fast core, which is a second generation algorithm. Now we have an advanced version of it, which is called the swift core. Okay, so you read up model integration or omics integrating algorithms, you will get like probably more than 50 algorithms out there. Okay, but the choice, sometimes people use more than 5 different algorithms for the same study. So depending upon the question you're trying to answer determines what particular integration algorithm you're trying to use. Okay, that is the most important takeaway from this slide. Okay, so the timing of this algorithm will depend upon the solver that you're using. Most of them are okay with using the Cplex or the Groby solver, which is pretty fast. These are MILP problems, mixed integer linear programming problems. And the last I used using this, yes, someone? Yeah, you can use IMAD, you can use MBA, you can depends upon the solver that you're using. GLPK will take a long amount of time to give you the optimized model. Okay, but then that is not the important thing. You have to see what is the question you're trying to answer. Okay, the most, if you want to build a model which is the exact replica of your data, then your gimme will not be the choice of algorithm. Gimme will require an objective function. What is it? If you say you're building a model for microorganism, your objective function is biomass, it will keep that objective function as the sole objective function. Any reaction that is fulfilling my objective function will be included in the model. Those reactions that are not supporting the biomass will be excluded. Okay, so that depends upon what is your question. However, if IMAT and MBA is also slightly tricky because you have to have a bit of domain knowledge. IMAT doesn't require domain knowledge, it only requires knowledge about the set of reactions that are medium and low expressed, that you anyways get from your data. Okay, you put your threshold, you divide it into 2 columns, feed it into IMAT using probably Cplex, Groovy, or whatever the newest solver from Stanford team has just been released. Within probably less than 10 minutes, you get your model. Size ranging from 7,000 to 8,000 reaction. Okay, so that part is easy. Difficult is matching up the phenotype of the data to that of your model. What is my model answering? Whether it is able to perform the same set of functions for which you are building the model for. Okay. Okay. So although we talk about automation using semi-automated methods to build models or any kind of computing environment, certain steps, I mean, it's slightly difficult to automate only because you need a bit of reading, you need a bit of domain expert. Okay, if you're building a model for kidney, you have to have transport proteins that are specific to kidney. Okay, you have to read a bit of literature. Probably omics will give you certain idea on that. Okay, then yeah. And then of course these are you also want to know, because you are building for microorganisms, it is pretty easy. You have just 2 subcellular locations. Whereas if you go for multicellular organisms, which has around more than 10 subcellular, meaning cytoplasm, Golgi, endoplasmic reticulum, mitochondria, nucleus, all of these put together, that particular location where this reaction is occurring, that information is slightly tricky to, even omics will not give you this. Okay, you have to read up a bit of literature. There are certain algorithms which is called p-sort, and then there are certain wet lab experiments which get released as to the tagging proteins to give you where this particular reaction is getting localized. All of that information can feed into this model and help in giving you the, it's just cutting out your errors. It's not that if you do not have that information, your model is bad. Okay, bad models can also give you good answers. Okay, it depends upon the type of questions that you're answering. Okay, so yeah, we move on to the next one. Okay, so that was about the different algorithms, certain exceptions, the takeaways, points that you need to remember when you are applying whatever algorithm that you want to. Okay, I'll give you certain examples as to how you can use omics data to derive a model of interest. I give you examples of 2 cancer models and one. I was a bit reluctant whether I should give this particular set of slides or not, but whenever we talk about diseases, we need to have a control. What is the healthy model that everyone is interested? When you are comparing diseases to that of the healthy, what is the healthy version? So I have a set of slides where we developed something called a healthy model. How is the human body performing when it is in a complete healthy state? Okay, I'll give you that example as well. And then of course we use that healthy model to derive certain disease models as well. Okay. Yes. It's all relative. So when you are designing your study, when you are saying this is my disease, let's say colorectal CRC, and this is my healthy, you need to design certain inclusion exclusion criteria which will fit only for the data and your particular study. If you look at the worldwide view, will not stand true, right? The same goes for a lot of these microbial studies. We see a huge amount of link between the gut microbes to that of the disorders. It could be metabolic disorders, it could be neurological disorders, but we see that link, gut microbes impacting the human health majorly because of the gut-brain axis. Okay, so when you study the impact of microbes to that of the human organs, you choose a bunch of organisms. You still do not know what is the totality of microorganisms that are inhabiting your gut, your various other locations. So how sure are you that these are the set of microorganisms and this is my community that is okay with this kind of cell type? Again, relativity. Disease type and your inclusion exclusion criteria, what exactly you have excluded. For me, healthy is just the absence of the disease. I am okay with CRC. I'm exactly, yes, yes, we have that same set of problems every time we are designing a study and we are trying to model or whatever, not exactly model but various other statistical way of determining the importance of the data. What exactly is healthy? The final conclusion, the basic set of inclusion exclusion criteria, but it may have, let's say for any kind of non-diabetic person, that can be healthy and diabetes is your disease. Doesn't mean that the non-diabetic will not have any other diseases. It's all relative. Healthy is a huge topic of debate. And yes, yeah, so I will not. Okay, I want to see if I am building a model to understand the pathology or the underlying biochemical basis of the disease, I will try to exclude as many risk factors as possible. Okay, but then to get that kind of data is very difficult. Probably from literature, but then you have to spend a lot of time reading and acquiring the data. Most of the omics data, if you look into the inclusion exclusion criteria of those studies, you will see what the kind of healthy profiles they have actually taken from. See, you can, again, it depends upon the question you are trying to ask. If you want to see the impact of these set of criteria on your final output, definitely include them or create a model that is very specific to that particular inclusion exclusion criteria. Yeah, but then the silent phenotype is again a slightly tricky question. Do you have enough data for those kind of studies? That would be the first thing that you will see. And how much of literature is out there for such, so that, because most of the time it is difficult to experimentally validate your simulations. You have to refer to the literature, whatever my model is predicting, does it make sense or not? So is there enough amount of evidence in the literature to support your computational predictions? Okay, so yeah, lot of bit of prerequisites also before you start the study. Yes. Hmm. Yeah. Yes. Unfortunately, but yes. Yes. Done? Okay. Sorry about this figure. The test case that I want to show you for using omics, deriving omics specific model from recon, I've chosen the example of cancer for the retina, which is called retinoblastoma. Not very abundant in Western population. In fact, even within India is not that high. It's a childhood tumor. You will see different stratification of the disease. This is the most aggressive form of the tumor. And most of the time, the solution to it is removal of the eye completely. Okay. So the question that we wanted to ask is, since we have data specific to the subtypes of the patients, okay, can we now use metabolic modeling to answer certain questions? Basically, can we now come up with certain biochemical understanding that is specific to the RB? Using RB as a short form of retinoblastoma, okay, biochemical understanding of RB to that of the normal. Okay, in this case, we got the normal data as well from comparatively healthy patients. Okay, so these are the different objectives for which the study was designed. First one is to build models specific to healthy, specific to RB. And then of course it has to be retina specific. We cannot do away with the whole recon or a whole human body. We have to have a retina or a organ specific model or a cell type specific model. And then can we come across and identify the biochemical basis that can then help in stratifying the patient groups. And then if possible, whatever our model is predicting, can we at least do some kind of experimental validation to show the model is kind of believable at this stage. And then of course, as every disease study has, can we predict novel drug targets and therapeutics? Okay. Yeah. So you start with recon. So recon at this stage we treat it as a library of metabolic reactions specific for human body. From that library, can I use the data? Okay. This was given to us generously from Shankar Netralaya, which is in Chennai. The vision research unit of that particular institute, okay, gave us the data. The transcriptomic data specific to the retinoblastoma and that of the healthy counterpart. Okay, and I used different kinds of model extracting or the model building algorithms using the data. I had my important question was which particular algorithm cut to which particular model is actually capturing the majority of the data. Okay, so that was, we end up with a preliminary reaction network or a model at this stage, and then we tested whether it is able to capture a lot of the data functionalities. It turns out in order to be specifically saying this is a retina model, you have to devise or formulate objectives around retina. Again, a bit of studying of the literature, we had to identify functions specific to the retina. At this stage, we did not only do positive controls, we also had to go for a negative control. What are the functions or the tasks that a retina functions as well as do not functions? And that's how the model was also tested for. Okay? And then we also used a lot of environmental constraints, basically nutrient media, under which these models were tested for. And then a lot of iterations were done, and then finally we ended up with models which are fairly reasonably capturing whatever the functions we asked it to perform as well as the data. Okay. So this is the plot that I'm showing you. On the x-axis, on the y-axis are the different metabolic functions or objectives which we formulated and the models were tested for. And each column that you see are the different subtypes of the data types that were given to us representing the patient stratified data. And we build models specific to the patients and asked, under the retinoblastoma compared to that of the healthy, which functions are performed and which functions are not performed. Okay, that's the only thing that we asked and wanted to check on the preliminary level. What we got information was pretty interesting because we saw that one of the energy precursors for the cell was completely different between the healthy and that of the cancer type. Okay, while the cancer models majorly used amino acids, the normal cells were basically depending upon the carbohydrates. Okay, the idea was, can we use or design certain therapeutic strategy that can flip it? Can we use something that can flip the usage of amino acids to that of the carbohydrate? Okay, so that hinting you on the therapeutic strategy that can flip, and you can do a number of simulations pertaining to that as well. The second interesting finding that we saw was one of the metabolite very important for the formation of your cell membrane is the cholesterol. It is a derived lipid. You will require cholesterol for maintaining your redox potential. Okay. However, cancer cells were not synthesizing cholesterol to that much extent as the normal cells were doing. And why was it doing that? We saw that one of the cofactors, so when you have reactions, there are certain cofactors that are utilized in order to furnish that particular reaction to its full capacity. These are called NADH, FADH2. These are redox potentials. Now, synthesis of cholesterol is a multi-step reaction, and most of these steps required the cofactor called NADPH. Why the cancer cell was not doing it? Because it wanted to preserve its NADPH reservoir. Why was it preserving it? Went back into literature, studied a bit on it. and then came to a conclusion by preserving your redox potential, you are not highlighting the those mechanisms which will come and kill the cells. So whenever there is a disbalance in your redox potential, there are certain pathways that are getting triggered, meaning that something is wrong. Now you have to come and kill this cell. If you cover it up, do not use your NADPH, you're never getting recognized by the protective mechanisms of the cell. Okay? So that was one mechanism that the cancer was adopting. Another thing that we saw was synthesis of fatty acids. We were not aware, it was one of our predictions when we were talking to our clinical partners, that we said that these cells are very specific to the kind of fatty acids that they are synthesizing for. And then the clinical partners advised us that this is a very, the same feature that we see in our patient groups as well. When we stratify our patients, they are based upon the kind of fatty acids that this particular patient type is synthesizing. Okay? So that was a big achievement for us, that we were able to capture a lot of biochemical basis for the cancer versus healthy. Okay, then we did a bit of experimental validation, not me, my collaborators. Okay, and they did metabolomics. So we developed the model, we ran the model, and the model gave a set of metabolites that are secreted out. Okay, this is what we call as a secretome. And that is what we tested for. Like now when you test it experimentally, do we get the same set of metabolites that is actually coming out from the cell, that versus of the model? And we were able to capture that yes, whatever the model was predicting is actually being seen on the experimental side as well. Then we used another algorithm which is called FastSL, which is collaborator that is coming from our collaborator's lab in IIT Madras. So he tested for knockdown of reaction in pairs. So when you knock down them together, you kill the cancer cell, but you do not harm the healthy cell. Okay, and that's what we call, and we identified certain transport proteins when you knock them together, that is with subdivision in the terms of nucleoside transporters and lipid transporters. Knocking them together is killing the cancer, it is not harming the healthy cell. Okay, so these were certain interesting aspects that we saw from the retinoblastoma study. Any questions or all okay? Is it boring? Yeah, I know, I get this a lot. Metabolism is really boring. Yeah. Okay, so we move on to the next test case, which is a colorectal cancer type. I knew the RB will be slightly heavy. I have very few slides to CRC. Please bear with me. Okay. This one is slightly interesting because we integrated one of the microbial models, a representative of the human gut bacteria, which is called the Enterococcus durans, E. durans. We built the metabolic model of E. durans, we built the metabolic model of healthy colon, build metabolic model of CRC colon, all coming from the human protein atlas data. So this is the proteomics, that was retinoblastoma transcriptomics, this is proteomics study. We also incorporate a bit of metabolomics data when it was available then, integrated everything onto the Recon 3D, derived our specific cell types, and wanted to understand how is the metabolism in CRC different from that of the normal cell type. And again, quickly I'll finish it off. Yes. Yes, oh yes, in fact more than 1,000. There was quickly and answering easily because this was available with our experimental collaborators. And however, when you want to design a bunch of bacterium, it is slightly difficult to experimentally grow them. You have a platform coming from my past lab, University of Luxembourg. They have devised a platform called Humix, where you can actually grow the bunch of gut bacteria. You can also grow your host cells and see the interaction together. That kind of methodology was somehow not available here. Whatever was available, whatever data was available, we tried to build a model around it. Is it the fact that this yeah, it is there. Yeah. No, Edurance is there. Edurance is a very normal bacteria. What we have seen in the literature that edurance tend to exert protective effects in the colorectal cancer. That was the initial question that we wanted to answer. But then as we went down the line, we realized studying in isolation doesn't make sense. If you want to know what is the protective effects of edurance, you need to have the collaborating partners within the gut. Okay, so that's why the results that I'll show you, we could not come across a result which is very specific to the bacteria. We were able to identify the protective effects within the bacteria, mostly at the levels of folate metabolism. But when we integrated into the colorectal cancer model, that effect was somehow losing. So whatever it was exerting, protective, harmful, or whatever, it can only exert when the other collaborating partners are there. Not only in terms of bacteria, you need to have certain fungi models as well. That's how the entire ecosystem is built. Okay, so these are again giving you the highlights that we identified pathways which were validated in the literature, that yes, colorectal cancer utilized or overutilized these pathways, mostly in terms of nucleotide metabolism, purine and pyrimidine, and a bit of fatty acid and branch chain amino acid metabolism. The reds that you see here are overutilization by the colorectal cancer models, and these are kind of helping the cells to proliferate at a much faster rate compared to the normal cells. This was experimentally validated, and I'm showing you only the computational part. The other part of the study was growing these cell lines and seeing whatever is happening here, whether it is matching onto the real world scenario or not. So this was actually validated as well. Okay, fine. Too much of modeling. Want to take a minute break, or you want me to continue? But this is the last cancer that you will see here. From here onwards, I'm showing you the normal cells, all happy things. Correct. Yeah. One of the aims that we had in the study was whatever the model was predicting had to be experimental validated. So how are you going to validate the bunch of bacteria growing together in association with your host cells? So that setup has to be there. Okay. So this kind of setup already existed with our collaborators, and that's the thought was whatever they had, let's just model it up. Okay. Okay. I can see like energy levels are going down. Okay. Shall we take a few minutes break? No? Shall I continue? As I said in the beginning, whenever we discuss disease, we require a relative healthy. The idea came and we started building something called a healthy model, and pretty extensive modeling in terms of methodology went in. A humongous amount of data sets went in. Okay, we started out with Recon 3D. We used proteomic information, we used metabolomics information, we used transcriptomics information, we used literature information, and a lot of other physiological data that was out there, integrated everything. And this time we were not building a generic model, we were interested in building organ specific models. Now when I say organs, what we ended up having is the most abundant cell type in that particular organ. Let's say for liver, we took only hepatocytes, and we termed it as the organ model for liver. Okay, and we ended up having 32 organ models integrated all together using the biofluid information that is out there. So we build it again gender specific. We built whole body organ resolved gender specific metabolic models for males and females. Okay, that's the typical framework that was used, and this is, yeah. So if you zoom in, you can see that 32 organs. We also have certain blood cell types. Okay, and unique about this particular model versus what was published previous, there are organ specific models. There are organ specific models that are integrated only at the level of blood. Okay, we integrated it at different biofluid information. So all of this integration that you see is still not completed. So the human body is just not this, it is more than this, but we were able to achieve to some extent a better model compared to what existed at that time. Okay. So again, interesting feature is the idea was to come up with a whole body model and then integrate the entire gut microbiome. Again, we have only taken the bacterial component, we have not taken the fungi part. Okay. So if you zoom in, for the small intestine, we had the lumen, and inside the lumen, we actually had the bacterial model. It's a pretty humongous model. You can see the size, that's the level of reactions. Okay. And that is only for the human component. If you integrate the microbial component, it is pretty large. Okay. Yes, yes. So that's what we said, these are not perfect. We attempted it, we tried achieving some parts of the human behavior and we were able to capture it, but this is not the totality. Okay, it is not even metabolism complete. Human body is just not 32 organs, it's much more than that. And even when you are saying one particular organ, we are taking only one cell type. But even with that, we were able to capture the BMR component. We simulated it on fully fed state, starvation state. What happens when the gut microbiome integrates and what is the impact on that? So few of those results were validated. Yes, 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 yes. Again, this is only metabolism. We did not even come across the non-metabolic. No, we did have. So even before going into literature, we had a series of organ function tests. You remember building Recon 3D, we had around 100 organ specific tests. We built upon that script. We had close to probably a double of that organ specific functions. Again, both positive and negative controls. Functions that the organ should do and functions that the organ shouldn't do. These are the simple biochemical tests that we asked the model to perform, and it performed. So yeah, just to give you, we have certain, yeah. So we started out with models that were already pre-existing, the ones that were constructed in a bottom-up fashion. That was for the small intestine, the adipocytes, and the liver. Liver is one of the most heavily utilized genome scale model that is out there. Okay, this is coming from the German group, and that is the model that we also integrated. And then we had certain the blood cell types. Okay, and certain gender specific organs, integrated it all together. And I'll show you what we did. One thing was, there are certain disorders either happening at one, so when you have your metabolic pathways and they are not functioning well, so you have deficiency in one enzyme, or it could be deficiency in multiple enzymes. Okay, those are the disorder which we call as the inherited metabolic diseases. Why inherited? Because they run within a family. Okay, we had that information prior. What are the close to 60 disorders and what are the genes that are getting affected, and we simulated that onto this particular whole body models, and we were able to see that whatever biomarkers are being predicted by this particular model was close, the predictions were not that off. Okay, it's again, it's not 10% correct and accurate, it's not that off compared to its counterparts. Okay, something else that this was very fascinating for me, because when we talk about integrating models and integrating so many different organ models all together, what we see within the human body is there are certain metabolic pathways that are operated in close collaboration with other organs. Typical example that you see is that of a Cori cycle, or the glucose alanine cycle, that is done in collaboration with the liver and the skeletal muscle. So what happens is glucose is getting fed from the liver to that of the skeletal muscle. It oxidizes it and leaves out lactate, which is taken up by the liver. And again, using gluconeogenesis, it synthesizes glucose again. Okay, so that's a cycle that happens. Many such cycles occur within the human body, and we were able to capture these phenomena, the inter-organ crosstalk at the level of metabolism. Okay, that was also nicely captured onto this particular model. Okay, and the last thing that I want to show here is, again, we integrated a bunch of metagenomic information at the level of gut microbes. Here we have only taken the bacterial component. integrated it together, and we saw that these microbes, typically the ones that are highlighted in this column, are very sufficient to synthesize something called neurotransmitters. Okay? The highlight of this particular slide is that this particular model will then be used very effectively to study the brain-gut axis to see the impact of microbes to various human organs under disease conditions. Okay, but the major objective, somehow I did not show it here, the major objective of this study was to capture the normal physiology of the human body. Okay, if you read that paper, it gives in details what are the different normal physiological functions that we were able to capture. Of course, the inter-organ crosstalk is one of the biggest factor that we captured, but we were also capturing various other factors. How much do we have? Okay, so I have a bunch of slides, but I'll try to brush it up pretty fast. Now, the question is that I have been saying from the very beginning, talk about diseases, it's just not metabolism. You have to study at the totality. Can we go beyond metabolism? What I mean is, what are the different gene regulatory cascades? What are the different protein signaling information that can be integrated all together in order to study the totality? And that is what we call as a whole cell model. Okay? The origin of the metabolic models or the genome scale models came from the genome annotation data that you see here in the late 90s. So because there was the annotation data, you built the first genome scale metabolic model of the corresponding organisms, the first whole cell model which actually integrated transcription, translation and metabolism put together. That was done for this Mycobacterium genitalium. This was a Cell paper released in 2012. Pretty heavy paper because the integration methodology was very complex, and you need to remember that when we talk about integrating, these processes are operating at different time scales. So how are you going to integrate all these processes that are molecular processes operating at different time scales to make sense of the final results? Okay, but this paper captured to some extent really good results for this particular organism. Okay, then in this particular era, we are seeing certain whole cell models. These are again at the level of bacterium, and then we also have for the yeast, which is a eukaryotic organism. Okay, but then we do have challenges, as I have been saying, collecting data, okay, but integration of the data for completely different molecular processes will be slightly tricky. Okay, this paper utilized something called coupling constraints, okay, in order to build the models. Okay, so that is one way of showing the future outlook as to how you can go beyond metabolism in order to study different processes. But other than that, when you're talking about an organ, you cannot do away with just one single cell type. How we did for the Harvey-Harveta model, the whole body model, we took only one cell type, but that's not how it is happening. You have to see the complete, or at least the top 5 cell types that are there for that particular organ. Okay, so that you can call it actually an organ specific model. Okay, so and then finally the last slide that I want to highlight here is the availability of something called the Human Pathology Atlas. Okay, this is coming from the same group as the Human Protein Atlas. These people, for the very first time, they utilize different data types, they build your metabolic models, but other than that, they also built a lot of these transcriptional regulatory networks and protein-protein interaction networks. But other than that, they also identified the prognostic value of the genetic signatures, okay, under different cancer types. They simulated 17 different cancer types. Okay, what is the extent and for which the genes can be used as a prognostic markers, and they have used the PFS curves. Okay, more about this in the next slide. Yeah, so what they have done is giving you 2 set of prognostic markers, something called favorable and unfavorable. Genes whose higher expression will favor in the, not for the cancer, but for the healthy type, and unfavorable, the under expression. Okay, that's what they give for the 17 different cancer types. And this is again kind of highlighting how we need to look at the genome scale models with that of a clinical angle. okay, in order to be used directly within the drug discovery and development pipeline, you have to have these kind of features. But then again, using the methodologies that have been used for building genome scale models, you have to have more advanced and more sophisticated methods in order to be in order to use these models in the D&D pipeline. Okay, again, these across the 17 cancer types, these are the top metabolic pathways that were highlighted. Nothing strange, because we have been hearing that a lot of lipid profiles are actually being used up as markers of the cancer. Just to show you. Okay, so that brings to my last, again, a text heavy slide. Sorry for this, but I've just given you 1 sentences for each study that I've shown. Okay, you can use a lot of omics data, a lot of literature domain experts, and use something called Recon 3D. It's upon you. Recon 3D is one of the human library of human reactions. There are different versions of it as well. Depending upon what you're trying to answer, you can use it and constrain it as per the data or for the environment under which you are studying, in order to make meaningful predictions for the disease type that you are trying to simulate for. Okay, and one thing interesting is, if possible, there should be a integration between the modeling field as well as the experimentalist in order to support one another. Okay, and then lastly, as I said before, these genome scale models have to be tailored with more sophisticated modeling approaches for them to be adopted in the D&D pipeline. And what will happen is, once you reach to the level of clinics, I think this part is something that Dr. Rukmini will take in more extensively, is you can make much more meaningful impact to the lives of those participants who are participating in your disease research area. Okay, that's it. So most of the studies, again, it's not me, it was a huge team of students and various other collaborators came together and did all of this. And the study that was the colorectal cancer and the retinoblastoma, this was done in IIT Madras. The support came from DST Inspire Faculty Fellowship. And whereas the Harvey-Harveta model was partly my PhD advisor in association with her. And yeah, so thanks for all the students in the team who came together and put forward this nice piece of work. And then, thank you. I'm ready to take in questions. That could be the starting level, but then see, this entire cascade of genes to level of your proteins to that of the reactions, it's not how do I say, it's not one way. We see more information from the level of metabolites or reactions, feedback loop to that of the genetic expression as well. Okay, so that directional thinking has actually changed. It's not like that. We see a lot of feedback and feed forward loop between these molecular processes.